Good morning, everybody. Thank you so much for being here this morning. I'm going to talk today about some of the work we've done in the last year, focusing on medications in milk. And Tina nicely set up our MPRINT study. But before I talk about that, I just want to sort of remind you all, we all know that breast milk is liquid gold and is essential for the infant's development in the first few months. I want to just focus on the fact that milk is a biological interface, and in addition to all the incredible nutrients and micro and macronutrients available in milk, we also have microbes and all the exogenous things that a mother intakes, whether it be drugs or environmental chemicals, and then all of those things have metabolites. Right, so the microbes produce metabolites. We have endogenous metabolites produced by the mother, and then, of course, the area that we're all interested in are drugs and food and all of the metabolites that those substances produce. So I'd like to sort of, introduce a paradigm shift in understanding breast milk. We have a very good way of determining, relative infant dose, looking at pharmacokinetics, and estimating how much of a drug an infant might ingest while they're breastfeeding. But I'd like to also have you remember that there are a lot of effects of these exogenous compounds on the breast milk itself and on both the microbiome of the breast milk. There's a specific microbiome within milk, and the metabolome, all the metabolites that are present in the milk. And how does that impact the infant's immune development and their overall development of health as they are growing in this very dynamic period of time, particularly between birth and 1 to 3 years when the microbiome is developing. So as Tina mentioned, we did a lot of untargeted metabolomics work, and for those of you who aren't too familiar with untargeted metabolomics, it's different than targeted work. It is less quantitative, but it is hypothesis generating. So it is a tool for discovery of new molecules, and we have discovered many new previously unknown metabolites. It's an important tool to understand biological function. It's the very end of, from the gene to the protein to the metabolite, and it's very sensitive technology. Now, the only issue is that we can get all of these peaks and we get all of these, potential identifiers, but the challenge is in actually annotating all of these chemicals. So we have a lot of dark matter, and our trainees spend a lot of time in the dark matter. And I'm going to show you a little bit of how we're trying to manage that dark matter to identify what these peaks actually are and what these compounds actually are. So as Tina alluded to, we, through her incredible milk biorepository, we had access to milk samples from 25 unique individuals. From her human milk biorepository, we subjected these to LCMSMS, and our talented postdoc Nina was able to then apply, she created what we're calling the GNPS drug library. So this is a publicly available resource of mass spectrometry data that has been annotated for drugs and drug metabolites. And we just recently published this in Nature Communications. And we looked at drugs that were present in the milk. And what we found was that, sort of no surprise, as Tina has shown, most of the drugs that we saw in the milk were antidepressants, followed by analgesics, and then antibiotics. And so we focused this analysis, which is preliminary, on the SSRIs. And I'd like to also point out that where our targeted assays fall short a little bit is in looking at metabolites. Because as you all know, when you do targeted analysis, you need standards, and we don't have standards for all these metabolites. But with untargeted metabolomics, we can actually interpolate those metabolites based on known chemical changes that happen in metabolism. And so Nina has been able to expand the drug library, and we're working on Drug Library 2.0 to include more metabolites and analogs that will then provide us more information about drugs. So without the metabolites and analogs, we only observed 83 medications, but that improved to 97 by including the metabolites and analogs. And I'll show you an example here with mesalamine, the common drug used in IBD. Initially, if you look on the left-hand panel, we were not able to detect anybody taking mesalamine, despite the fact that 22 of those women reported taking it. When we included the microbially-mediated metabolite 5-acetyl N-acetyl 5-ASA, we were able to show that indeed 19 women were exposed to mesalamine. And there were actually 3 who didn't report taking it, but we did detect the metabolite. So sometimes, as you all know, when you do clinical research, the metadata doesn't always correspond with what we actually see in the sample. And then this is just an example of 4 drugs that we only saw as metabolites in the breast milk. And so again, we would have missed this, the fact that these women were exposed to these drugs if we weren't also looking at the metabolites. So just a variety of different drugs and their associated metabolites. So what about the effects on the milk metabolome? We looked at all of these different chemical entities that we found in the milk and that were shifted by drugs. The majority, as we mentioned, of the drugs were antibiotics and antidepressants. And so we focused initially on looking at the metabolome's effect on these agents. It looks like antibiotics are decreasing the diversity of the metabolome, whereas antidepressants are a little bit more of a mixed picture, a little bit of decreasing of the metabolome diversity, and then a little bit some of the metabolites that are present in those women who are taking antidepressants and those who aren't. So we identified 10,000 MSMS spectra, but as you can see, only about 700 of those are annotated. So this is, again, why we have so much dark matter. So I'm going to show you some of the metabolites that we saw that were shifted, but there might be many more that we are continually working on to annotate. 45 of those 707 were associated with antidepressants, and most of them were seen in those not exposed to antibiotics. So thinking about it, that these metabolites are in lower amounts in those women taking antidepressants. So a carnitine derivative. Carnitine is, known for transporting fatty acids into the mitochondria for energy production, the di and tripeptides, which are likely involved in protein metabolism, and then some lipid compounds. So we're continuing to work on this data set. We're going to be looking more into the antidepressants and those metabolites that were increased or enhanced in those women who were taking antidepressants. And we're going to be continuing to work on increasing the annotation of those and using additional computational strategies such as reverse cosine search to find additional novel drug metabolites. So now I want to switch gears and talk about that MPRINT clinical study that Tina nicely set up for us. So as she mentioned to you, we enrolled 40 women, 40 mother-infant dyads who were taking antibiotics and 40 matched controls. They all produced baseline samples, and then once the women who started antibiotics started taking antibiotics, we had matched controls provide the same samples of feces, milk, and skin, both feces and skin, both for the infants of the mothers, and then obviously milk samples from the mom. We subjected those to untargeted metabolomics and shotgun metagenomics for microbiome analysis. And then Jeremiah and Ray did the targeted antibiotic quantification. So in terms of the demographics of the population, they were pretty well matched. There was a slight trend in maternal BMI to be a little bit higher in the, a little bit higher in the antibiotic group, but that did not reach statistical significance. And as far as which antibiotics those women were taking, they were mostly on beta-lactam, so most of them were on amoxicillin or augmentin and Keflex, and then, with fewer amounts of the non-beta-lactam, such as Clinda and Doxy. So Ray and Jeremiah actually created a multiplex LCMS assay for us. And through that assay, we found that 90% of the milk samples showed the presence of antibiotics. And the concentrations varied pretty widely. There was not necessarily women weren't told exactly when to take their samples with respect to when they took their antibiotics. So that's partly, the reason why there's so much variability. But despite this, none of the, when calculating, the relative infant dose, they were more in the very low concentrations and didn't reach any sort of therapeutic concentrations in the infant. So I'm going to break up our results into different categories. So I'm going to talk about the impact on the human milk metabolome first. We saw a small impact by antibiotics on the milk metabolome. So looking at just this unsupervised PCA, there was a slight separation between both antibiotic group compared to baseline as well as compared to the no antibiotic group. And then when we did a supervised model, we did see significant separation between the women taking antibiotics versus no antibiotics as well as baseline. And we took the top 100 features with VIP scores greater than 1 and looked at what those metabolites were. One of the main metabolites that we saw that were enriched were acylcarnitines. And so again, we talked about acylcarnitines before, their role in energy metabolism and their role in shuttling fatty acids into the mitochondria. And then we did see some putative other microbially derived metabolites that were depleted in the milk metabolome. So moving on now, because one of our main questions for this study was, does indirect antibiotic administration cause changes to the infant microbiome? Because the microbiome is developing at this very critical time of infant development, and we know that direct infant antibiotics does impact the infant developing microbiome, but what about indirect antibiotic administration? And so Simone, one of our postdocs in the lab, did this microbiome analysis, and we did find that the infant alpha diversity did shift between baseline and no antibiotics and antibiotics. It increased, and this was likely due to the fact of age. So as infants develop, their diversity increases, right? As they are developing their microbiome, it's becoming more and more adult-like until it becomes more adult-like at age 3. And so it's likely the fact that the baseline samples were collected on average about 4 months prior to when we started collecting the samples for the antibiotic control and the control group. When looking at just a PCA plot, there was a significant shift in the microbiome distribution between the antibiotic group and the control. But this was considered, small compared to looking at the age effect and the delivery. So whether or not the mother delivered via vaginal or C-section. So keep that in mind, that it is a significant shift, but the amount of contribution of antibiotic was smaller than age or delivery. And it seemed to be that the effects were dependent on the type of antibiotic. So we had, as I showed you, most of the women were taking beta-lactam antibiotics. And so we looked at just those women, looking at both effect on alpha diversity on the women taking the beta-lactam antibiotics and including azithromycin as well. So amoxicillin, augmentin, Keflex, and azithromycin. And you'll see that the impact on alpha diversity was greatest in the women taking those antibiotics. And we also looked at the antibiotic effect, so looking at the log ratio of depleted taxa compared to enriched taxa, and those with the largest delta were the beta-lactam and azithromycin antibiotics. So we took that group of women and continued on our analysis. And so we looked at all of the bacteria that were depleted and enriched in response to indirect antibiotics in the infants. And in a supervised PLSDA model, we found approximately 250 different microbial species that were significantly shifted with antibiotics. And some taxa were depleted and some were enriched. And those that were depleted were those that were considered to be more beneficial bacteria. So Bacteroides and Bifido are both metabolized HMOs, and they are associated with immune system function, and Lactobacillus is associated with decreasing risk for infections. And then we also saw other bacteria that were enriched in those infants. We also, interestingly, saw a higher proportion of antimicrobial resistance genes in those infants that were indirectly exposed to antibiotics. And that was statistically significant, potentially posing a risk for the infant resistome in the future. Okay, now moving on to the infant fecal metabolome. So Kine is our main postdoc working on the MPRINT, many of our MPRINT studies, and she looked at all the metabolome data. Looking at unsupervised PCA, we saw a small shift in the metabolome of the infant due to antibiotics. And then when looking at a supervised PLSDA model, we saw a significant enrichment and depletion of the top 100 features of the metabolites. And you'll see that acylcarnitines showed up in the most amount in terms of the metabolite classes, followed by bile acids, dipeptides, amino acids, and N-isolipids. And looking at depleted and enriched classes, we saw some short and medium chain acylcarnitines depleted with antibiotics. And interestingly, about 31% of those were microbially mediated. Excuse me, I'm just gonna have some water. So MicrobeMast is a tool that we have that is pairs metabolites with microbial monocultures. So we can see if the metabolite has bacterial metabolism associated with, and 31% of these metabolites did, suggesting that those acylcarnitines were microbial metabolites. Again, suggesting that the antibiotics shifted those metabolites. We also found shifts in other metabolites, indole-3 acetic acid, which plays a role in intestinal and immune homeostasis. We looked at heme catabolism. So there was a shift in increased urobilin and decreased stercobilin, suggesting that the enzyme that converts that was depleted. And then finally, enrichment of several N-isolipids. And then interestingly, looking at clinical outcomes in a subset of infants, we found higher BMI at 2 years with antibiotics. And this is known when children get direct antibiotics. So in summary, I just wanted to show you data that we think shows the importance of the role of drugs and drug metabolites in altering human milk composition. Antibiotics do get transferred to the infant via human milk during maternal treatment. Maternal antibiotic treatment does seem to have a small but observable impact on the infant gut microbiome and metabolome. And in addition to its important nutritional role, milk contains a complex array of bioactive components, including microorganisms and their associated metabolites. And, really metabolites are the language of the microbiome, and we're learning more and more of the role of these metabolites in physiology and health. And I think it's important to look at this in the context of the maternal-infant dyad and the developing infant microbiome. I just want to acknowledge all the people who've done the work on this project. Kine, our main postdoc in my lab, Simona and Nina, very talented postdocs in Peter's lab, and just a shout out to Peter for creating all the infrastructure that makes all these studies happen, and Rob Knight, who created the infrastructure for us to do the microbiome analysis. And of course, my incredible MPRINT CET team. Thank you.